gene, which removes the primers. We also use the gross gel electrophoresis, which separates the macromolecules such as DNA, RNA, and proteins. So what happens within the process is throughout the gel electrophoresis, the DNA is inserted here, and then once the voltage at 250 occurs, the DNA will slowly move towards the cathode because DNA is already negatively charged, but since there's an electric force, it will start to move towards the positive end. The gel gross electrophorese separates by size and mass. So the smaller the macromolecule, the faster it will journey to the cathode, the positive side. So specifically, the amplicon PCR amplifies the 16S ribosomal RNA gene, which contains the type of bacteria present in the gut microbiome of the selected animal we used in the experiment. In order to correctly emphasize the gene, we first denaturize the DNA strand at 96 degrees Celsius. This causes the two DNA strands to separate. Next, we cool the DNA strands, or we anneal the DNA strands at 54 degrees Celsius. And lastly, to extend the DNA strand, we raise the temperature back to 72 degrees Celsius to allow the tag polymers to attach the new single-stranded DNA. This process is repeated 25 to 30 times in order to get a sufficient amount of our DNA sample. Continuously, the PCR cleanup, which we used in our materials, is the process of disposing the PCR primers, DNA polymers, nucleotides, and PCR buffers from the DNA strand. We do this because the PCR primers will cause, within the next steps, the PCR indexing to not work. Basically in this step, we're re-isolating the DNA as we did in week 1. First, we insert the PCR reaction into a little capsule this big with a bunch of magnetic beads. So the magnetic beads can connect to the PCR product. The PCR reagents that are within the sample are diluted with 80% ethanol twice. But this allows the DNA to stay attached to the magnetic beads. Once we flush the PCR reagents, we insert the capsule into a holder, and it has a magnet at the bottom, so the beads immediately attach to the end. Finally, to detach the DNA from the magnetic beads, we insert Trish buffer. This step is required in order to have DNA in its most pure form. Continuously in PCR indexing, right here, this is very similar to the amplification process of 16S ribosomal R gene. The DNA strands are again denaturized, annealed, and extended. Once it is extended, the special DNA sequences, the tag polymers, So the DNA strands are denaturized, annealed, and extended, and once it is extended, special DNA sequences attach to the 3 end of the DNA strand, which is the end, not the 5, because the 5 contains the tags which identify the DNA strands throughout the process, so they do sequenize in the end of our lab. Lastly, the quantification of DNA is processed through the fluorometric assay. The fluorometric assay is used in this step because it is more specific and targets our sample a lot more. So the fluorometric assay can read RNA, proteins, and DNA, which the UV spectrometer cannot. The technique is used to identify any impurities we still have within our DNA, such as buffers or free nucleotides. The fluorometric assay uses a dye that attaches to the DNA to allow light to pass through and measure the amount of DNA contained. the dye will only connect to the RNA, not the, will only connect to the DNA, but not the RNA or the proteins. So here, and then up here, I have my methods from week 1 through week 4. So in week 1, our goal was to isolate the DNA, and then we concentrated the DNA sample with the 10-fold. During this week, we used the key column as well as the Zymo kit in order to isolate the DNA. In week 2, during the week, we used the UV spectrometer in order to determine if our sample was contaminated with unwanted proteins. This week, we amplified the regions of the 16S ribosomal RNA, regions 3 and 4, which I stated earlier. And this allows the polymerase chain reaction, and using the polymerase chain reaction to determine the types of bacteria present in the animal gut microbiome. In week 3, we use the gel electrophorese to see the size and the mass of each macromolecule, DNA, proteins, RNA that are in our sample. We also perform PCR cleanup within this step to have the last purification of our DNA sample. Lastly, in week 4, we did several calculations. As you can see here, C1 to V1 is equivalent to C2 and V2. We also had to do many conversions using grams to liters, grams to moles, and then moles of liters to molarity as our final calculation, as our final unit calculation. Lastly, we use the conversions of millimeters to nanometers with 1 times 10 to the 9th. So besides the calculations, this was our last step to sequence the DNA and find out how much of our DNA was truly pure, concentrated, and found in the end. Okay. So here we have our dilution results for our PCR, positive and negative control. So the positive control, again, was our E. coli, and that allowed us to see that our results of our E. coli were not correct, but what we wanted or what we expected, not exactly pure, as it had, here are calculations, as it had 19.88 H2O in it, which means the rest was DNA sample. So here are our results. So the Kingdom classification results showed that the bacteria within our samples 1 and 2 were fairly similar, as both of them read about 10,000 and like 700. Next we have our order classification. Bacterioids were the highest on the sample, and then the chloristridiols were the second, and the first, the highest bacteria on this the highest microbiome on this chart was the chloristridiols. So Chloristium and bacteroids are both related to the bloodstream, so I will assume that a microbiome within the environment that each of our samples live in have digested within their system to cause a disorder of some kind within their bloodstream. Also within the class section, so Bacteroideae and Clostridia read at 3,000 and almost 2,000 for the Clostridia, and in our second sample, Clostridia read at almost 8,000. So from this, I concluded similarly that these results may be from their diet, as both of them have a somewhat vegetarian diet like grass and vegetables. And the last one, the phylum, bacterial, bacteroidetes and firmicutes were the top 2 on this sample and was in the top 3 of this sample. So the correlation between firmicutes and bacteroidetes is a gut microbiome that is correlated to obesity. So another assumption I have made is that the diet that they both consume is high fat protein, which so when firmicutes levels are high, the bacteroids fall. So I can assume that both of them had a somewhat high fat protein diet. In the end, a sufficient amount of scat DNA was extracted to allow us to compare the 2 organisms. And it showed that they were pretty similar. Within our results, it is pretty clear that they're 2 different organisms. They do live in different environments. But they do have similar correlations within their diet, as well as different they don't live in similar environments, but there are certain aspects of the environment which they live in that could correlate to their bloodstream, as I stated earlier. Overall, the gut microbiome experiment was successful, and I think we did a good job.